हेलो गाइस। आज है 7 तारीख अप्रैल का। और अभी हम लोग प्रिपेयर करने वाले हैं फोटोसिंथेसिस। उसको उसका एक वर्क दिए थे जहाँ पर हमने हाल ही एक्सपेरिमेंट्स के बारे में पढ़े थे। चलिए शुरू करते हैं। हाय वैशाली गावड़े, हाय। गुड मॉर्निंग वैशाली गावड़े, गुड मॉर्निंग। The synthesis of complex organic substances. रानी, हेलो रानी। कोलकाता में है यूनिवर्सिटी। So the हाँ, हमारा खाना हो गया। आप कहाँ से हो वैशाली? Photosynthesis is the process by which green plants use sunlight to make their own food. The food they make is glucose, a type of sugar. They take पुणे से। ठीक है, बढ़िया। Photosynthesis is the process by which green plants use sunlight to make their own food. The food they make is glucose, a type of sugar. They take in carbon dioxide from air and water from the soil. With the help of sunlight and chlorophyll, they convert these into glucose and oxygen. So the synthesis of complex organic substances, carbohydrates, by green parts of the plant in the presence of sunlight with the help of CO2 and H2O is called photosynthesis. It can be shown by the following equation, that is 6CO2 12H2O, 6 is H2O6 plus 6H2O plus 6O2. Photosynthesis is important due to two reasons. Photosynthesis is important due to two reasons. It is based on the food chain. All food on earth is directly or indirectly बोटानी पड़ता है। ठीक है वैशाली, बोटानी। It is the base of all the food chain. All the food on earth directly or indirectly comes from photosynthesis. Plants feed herbivores and herbivores feed carnivores. So it all starts from plants. It releases oxygen we breathe, is a byproduct of photosynthesis, without which life would not be impossible, would not be possible for animals or humans. To prove that starch is made only in green part of the plant, chlorophyll is necessary for photosynthesis. And to prove that starch or food in green parts of the leaf chlorophyll is necessary for photosynthesis. Take a variegated leaf, a leaf with both green and white part. The green part have a chlorophyll, the white part does not. The leaves contain a variegated leaf, they are shelley covered in black pepper, and once that was exposed to light, result photosynthesis occurs in the green parts of the leaves in the presence of sunlight. Result photosynthesis occurred in the green parts of the plant in the presence of sunlight. Half-leaf experiment, a part of a leaf that is enclosed in a test tube containing some KOH soaked cotton which absorbs carbon dioxide while the other half is exposed to air. The setup is then released in light for some time. When testing starts later in the two halves of the leaf is formed, it was found that the exposed part of the leaf tested positive for starch while the portion that was in test tube tested negative. So carbon dioxide was required for photosynthesis experiment. Boil the leaf in water to kill it. Boil it again in alcohol to remove the green color. Wash it and dip it in solution. Green part of the leaf turns blue-black. Starch is present. White parts do not change color. No starch. Half-leaf experiment. To show that carbon dioxide is essential for photosynthesis, take a healthy plotted leaf, keep it in the dark for 2 to 3 days to remove all the stored starch. Then insert one leaf still attached to the plant in a conical flask containing potassium hydroxide solution because KOH absorbs carbon dioxide from the air inside the flask. Another of the leaf stays outside, exposure to normal air with carbon dioxide, expose the plant to sunlight for a few hours, plug the leaf after sun boil to kill the cells, boil in alcohol to remove chlorophyll, rinse in warm water, test retardant solution, the half leaf of the flask with no CO2 does not turn blue-black, the other half turn the flask with CO2 turns blue-black. So the conclusion, CO2 was able to do photosynthesis and produce starch. Carbon dioxide necessary for photosynthesis. What is the aim of half-leaves to prove that carbon dioxide is not photosynthesis? What is the plant kept in darkness before to discharge the plant remove any pre-existing starch? Why is the potassium hydroxide used, which absorbs carbon dioxide from the air inside the flask? What happens on the half-leaf of the experiment? It does not turn blue-black with iodine because it does not dissolve carbon dioxide. So starch was formed. What happened to the half-leaf? The outside turns blue-black with iodine as it got CO2, so potassium occurred and starch was formed. What is the conclusion of half-leaf? CO2 is essential for photosynthesis. Stefan Hels. Stephen Hales, the forgotten pioneer. Stephen Hales was an English scientist. Stephen Hales was an English scientist. Okay. Plant absorb air through leaves. He suggested that important for CO2 and light photosynthesis in addition remains and he performed series of experiments that ensure that essential role of air in the growth of green plants. Plant key contribution plant absorb air through leaves. He suggested that plants take in some part of the air which we know today carbon dioxide through their leaves. Root absorb water. Root absorb water and sunlight is important for plant processes. Even though Hales did not directly prove photosynthesis, his idea laid the foundation for later discovery. He was among the first to treat plant as a living organism that interact with air, water and sunlight, a big shift in that time. He often called the father of plant physiology. Joseph Priestley, 1770. He placed a burning candle in a closed bell jar. After some time, the flame went out. The air was damaged. Then he put a mouse in a bell jar. It died due to lack of air. But when he added a green mint plant to the jar and kept in sunlight, the candle could burn again. The mouse survived longer. Conclusion: the plant restored the air that was damaged by the candle or the mouse. Later it was understood that plants release oxygen into the air. Priestley discovered that plants purify air, which was later linked to photosynthesis. What was the aim of Joseph Priestley? To find out how plants affect the air around them. What happened when Priestley placed a candle in a closed bell jar? The candle flame went out and after some time due to lack of oxygen. What happened when a mouse was placed? The mouse died due to suffocation, lack of oxygen. What change did Priestley observe when a mint was added? The candle burned again and mouse survived longer. What was Priestley's conclusion? Green plants restore the air. They release something later identified as oxygen necessary for breathing and burning. He showed that plants purify air by releasing oxygen, a key step toward understanding photosynthesis. What did Joseph Priestley demonstrated with his Benjar experiment? In Priestley experiment, what happened to the mouse kept alone in a closed jar? What was the result when both the plant and the mouse were kept in a closed jar with sunlight? What is released by the plant that helped the mouse and candle survive longer? What is the correct conclusion of Priestley experiment? Light is essential for photosynthesis. Carbon dioxide is green plants restore air quality. Jan Ingenhouse. In an elegant experiment, Priestley found what is released, Ingenhouse found when and how. He placed an aquatic plant like hydrilla in a transparent container filled with water. He placed aquatic plants like hydrilla in a transparent container filled with water. The container was kept in sunlight and he observed bubbles coming out from the green plants of the parts of the plant. No bubbles appeared when the plant was kept in darkness. Observation bubbles formed only in sunlight and formed green parts. The gas released was later confirmed to be oxygen. Sunlight is necessary for photosynthesis. Only green parts of the plant can carry out photosynthesis. Plant release oxygen during photosynthesis. Plant release oxygen during photosynthesis. In an elegant experiment with an aquatic plant, in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later he identified these bubbles to be oxygen. He showed that it is only the green parts of the plant that could release oxygen. He showed that sunlight is essential to the plant process that somehow purify the air fouled by burning candles or breathing animals. Julius von Sachs. Discovery of starch formation. He discovered that chlorophyll is found in special structure called chloroplast in plant cell. He discovered that chlorophyll is found in special structures called chloroplast in plant cell. He observed that when a green plant is exposed to sunlight, starch accumulates in the He observed that when a green plant is exposed to sunlight, starch accumulates in the green parts. When the plant is kept in darkness, starch is not formed. He provided the evidence of production of glucose when plants grow. He showed that product of photosynthesis as glucose which is stored as starch. His later study showed that green substance in plants, chlorophyll, is located in special bodies called chloroplast within plant cell. He found that green parts in the plant is where glucose is made and that glucose is usually stored as starch. Sachs proved that food starch is made in green parts of the plant through photosynthesis. Sachs proved that food starch is made in green parts of the plant through photosynthesis. He showed that product of photosynthesis is glucose. He showed that product of photosynthesis is glucose and the starch is kept at and the starch is in the chloroplast store made by chlorophyll. হ্যাঁ
তোমাদের নিচে পিছন দিকটা কাগজ নেই পাড়া 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 একটু ওটা ওটা ওই আয়নাটার পিছন দিকে কাগজ মেরে দিলে আবার ঠিক হয়ে যাবে মানে আবজা আবজাটা থাকবে না
না
আটা দিলে কি বিট বোর্ড দিয়ে মেরে দিলেও হয়ে যাবে তাই তো
হ্যাঁ
মানে ওই জন্য আবজা লাগছে মানে ওই নেই তো পিছনে ঢাকা শুধু কাজটা
হ্যাঁ আগেকার দিনে কাঁচ তো মানে আয়না তৈরি হতো একটা লোহা নিয়ে সেটাকে এত ঘষতো যেটা কাঁচে তৈরি হতো।
ওইটা কাঁচের পিছনে যেটা দেওয়া থাকে
না, এখন তো অন্যভাবে তৈরি হয়। না, আগেকার দিন আয়না তৈরি করতো ওই অ্যালুমিনিয়ামকে ঘষে 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 ঘষে। অ্যালুমিনিয়ামকে এত ঘষতো পুড়িয়ে যে ওটা তখন একটু চকচক, তাও আবঝা আয়নাই হতো, কিন্তু দেখা যেত না।
এখন কাঁচ তো তৈরি হয় বাড়ি থেকে নাকি?
হ্যাঁ।
কাঁচের গুঁড়ো কাঁচের দোকানে,
হ্যাঁ।
সব কোথায় যায় জানো?
কোথায়?
রোড গুলো তৈরি হয় রাস্তা,
হ্যাঁ,
ওর সাথে মেশায়। এই জন্য রাস্তাগুলো চকচক চকচক করে না,
ও
ওদের সাথে মিশিয়ে লোকা চেঙ্গে চলে যাবে সব। কাঁচের দোকানে কাঁচ কাটে তো,
হ্যাঁ।
কাঁচের গুঁড়ো করে তো, ওগুলো সব এই রাস্তার সাথে মেশায়।
Facts prove that food or starch is made up in green parts of the plant through photosynthesis. T.W. Engelmann, using a prism, he split the light into its spherical components, then illuminated a green alga, Cladopora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 revolution. He observed that bacteria is accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption of spectra through chlorophyll A and B. কি করেছিল টি.ডাব্লিউ এঙ্গেলম্যান? অ্যালগির একটা সেকশন কেটে ওটাকে So T.W. Engelmann, using a prism, he split lights into its spectral components and then illuminated a green alga, Cladophora, placed in suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. So, uh, uh, যে অক্সিজেন bacteria that love oxygen that goes into the part of that algae in the blue and red light area, and it is roughly the absorption spectrum of chlorophyll A and B. So blue and red light area is the most part of the photosynthesis where the algae took place. Blue and red light. Cornelius van Niel. Cornelius van Niel. General theory. He made a groundbreaking contribution by figuring out that general equation of photosynthesis based on work with bacteria. What he observed, he studied the purple and green sulfur bacteria which do not release oxygen during photosynthesis. He studied purple and green sulfur bacteria which do not release oxygen during photosynthesis. These bacteria used hydrogen sulfide instead of water and produced sulfur instead of oxygen. These bacteria used hydrogen sulfide instead of water and produce. He studied purple and green sulfur bacteria which do not release oxygen during photosynthesis. These bacteria used hydrogen sulfide instead of water and produce sulfur instead of oxygen. His observation: oxygen produced during photosynthesis does not come from carbon dioxide, it comes from the oxidation of water. General equation proposed by Van. Why is this important? Manley laid the foundation. Later experiment Robert Hill proved that oxygen comes from water, not from carbon dioxide. His conclusion. So जो उन्होंने बोला, he studied purple and green sulfur bacteria जो कि which do not release oxygen during photosynthesis. These bacteria used hydrogen sulfide H2S instead of water and produce sulfur instead of oxygen. Oxygen. His observation: oxygen produced during photosynthesis is not by carbon dioxide, it comes from oxidation of water H2O. General equation. Why is this important? When Neil laid the foundation for the experiment, proved that oxygen comes from water, not from CO2. Corners were near which organism to understand the mechanism of green and purple sulfur bacteria. 2. So the early experiment topic is over. Now we will move to the uh, next part, that is site for photosynthesis. The main topic starts from here, but before that we will revise. Okay. The synthesis of complex organic carbohydrates. Photosynthesis. What is photosynthesis? Photosynthesis is the process by which green plants use sunlight to make their own food. The food they make is glucose, a type of sugar. They take carbon dioxide from and water from the soil with the help of sunlight and they convert these into glucose and oxygen. Photosynthesis equation. This equation shows 6 molecules of carbon dioxide, 12 molecules of water, 1 molecule of glucose, 6 molecules of oxygen and 6 molecules of water extra. Okay, what is photosynthesis important? It is the basis of all the food chain. All the food on earth directly or indirectly comes from photosynthesis. Plants feed herbivores and herbivores feed carnivores, so it all starts from plants. It releases oxygen. The oxygen we breathe is a byproduct of photosynthesis. Without this, life would be not be possible for animals or humans. Take a vegetative leaf. Aim to prove that starch is made only in green parts of the plant, that is, chlorophyll is necessary for photosynthesis. The green parts have the chlorophyll, the white does do not. First, keep the plants in dark for 2 to 3 days. Remove any starch stored, then expose the plant to sunlight for few hours. Boil the leaf in water to kill it. Boil it again in alcohol to remove the green color. Wash it and dip in iodine solution. Green parts of the leaf turn blue-black. Starch is present. White parts do not change color. No starch. Conclusion: only the green parts of the leaf perform photosynthesis and produce starch. And so chlorophyll is essential for photosynthesis. Chlorophyll is essential for photosynthesis. Next is half-leaf experiment to prove that carbon dioxide is essential for photosynthesis. Take a healthy plant, keep it, remove it the starch, all the residue starch, and put it in a QH solution and one part of the leaf outside and normal airs. Then the starch shows. That means the other half stays outside the flash exposed to normal air with carbon dioxide. Plug the leaf and boil it in water to kill the cells. Boil in alcohol, freeze in warm water. Test with iodine solution. The half inside the flask with no CO2 does not turn blue black. The half outside does not turns blue black. Conclusion: the part of the leaf that got CO2 was able to do photosynthesis and produce starch. Half-leaf experiment to provide CO2. What is the aim of half-leaf experiment? To prove that carbon dioxide is essential for photosynthesis. Okay. Why is the plant kept in darkness before the experiment? To discharge the plant. Remove any pre-existing starch. Why is the potassium hydroxide used in the KOH? Absorb carbon dioxide from the air inside the flask. What happens when the half leaf of the total inside the flask? It does not turn blue black with iodine because it did not receive carbon dioxide, so no starch was formed. What happens with the half of the leaf outside the flask? It turns blue black with iodine as it got CO2, so photosynthesis occurred and starch was formed. What is the essential conclusion of the half-leaf experiment? CO2 is essential for photosynthesis. What is the main purpose of the half-leaf experiment? Why it is the paper? In the half-life experiment, potassium hydroxide is used to uh, absorb carbon dioxide. In half-life experiment, only one part of the leaf turns blue-black due to because carbon dioxide was available only to that part. Which of the following is not a necessary step before starting the half-life experiment? Necessary step is cutting the leaf into two halves. The half of the leaf inside flower does not turn blue black because it did not get carbon dioxide. Half-leaf experiment proved that carbon dioxide is essential for photosynthesis. Correct. Stephen Hales was an English scientist who made early contribution that plant absorb air through leaves, root absorb water, sunlight is important for plant process. Even though Hales did not directly prove photosynthesis, his idea laid the foundation of later discoveries. He among the first to treat plant as living organism that interacts with air, water and sunlight. He is the father of plant physiology. He placed a burning candle in a closed bell jar. After some time, the flame went out, the air was damaged. Then he put that mouse in a bell jar. It died due to lack of air. But when he added a green mint plant to the jar and kept in sunlight, the candle could burn again. The mouse survived longer. Conclusion: the plant restored the air that was damaged by the candle or the mouse. Later, it was understood that plant released oxygen into the air. Key points: briefly discuss that plants purify air, which was linked to photosynthesis. Flashcard: Joseph Priestley. What is the aim of Joseph Priestley? To find out how plants affect the air around them. What happened when Priestley out after some time due to lack of oxygen? What happened when a mouse was placed in a closed bell jar? The mouse died due to suffocation, lack of oxygen. What change did Priestley observe when a mint was added to the bell jar with mouse or candle? The candle burned again and the mouse survived longer. What was Priestley's conclusion from his experiment? Green plant restored the air. They released something necessary for breathing and burning. What is Priestley's contribution to photosynthesis? He showed that plants purified air by releasing oxygen and skipped towards the photosynthesis. Plants absorb Plants give up oxygen. In Priestley experiment, what was found in a closed jar? It died due to lack of oxygen. What was the result when both plant and the mouse were kept in a closed jar with sunlight? Mouse survived longer. What is released by the plant that helped the mouse and candle to survive longer? That is oxygen. What is the correct conclusion of Priestley experiment? That green plants restore air quality. Jan Lingenhaus. He placed aquatic plants in transparent water filled with water. The container he found that when the aquatic plant was kept in dark, it does not form bubbles. But when the aquatic plant was kept in sunlight, it formed bubbles and it is the release of oxygen, he concluded. Sunlight is necessary for photosynthesis. Only green parts of the plant can carry out photosynthesis. Photosynthesis released oxygen during photosynthesis. Simple lines to remember. Presley found what is released. Lingenhaus found when and how. Julius Bond Sachs. He discovered that chlorophyll is found in special structure called chloroplast in plant cell. He observed that when a green plant is exposed to sunlight, starch accumulates in green plants. When the plant is kept in a darkness, starch is not formed. Green parts of the plant make starch during photosynthesis. Glucose is the first product of the photosynthesis, which is later converted into starch and stored. Chlorophyll is essential for this process. Key contributions: Sack proved that food starch is made in green parts of the plant through photosynthesis. Cornelius van Nys. Let's explore Cornelius van Nil. He did photosynthesis and worked with bacteria. He studied purple and green sulfur bacteria which do not release oxygen. Instead of oxygen, they release hydrogen sulfide instead of water and produce sulfur instead of oxygen. The bacteria used hydrogen sulfide instead of water and released sulfur instead of oxygen. His observation on oxygen produced during photosynthesis does not come from carbon dioxide, it comes from oxidation of water. General equation: why is this important? Yes. Founders were studied which organism to understand the mechanism of green and sulfur bacteria. According to Van Nil, the source of oxygen released during photosynthesis is hydrogen sulfide. What was the significance of Van Nil? He proposed the general equation. He showed that O2 comes no. He showed the emergence of sunlight. He proposed the general equation for photosynthesis. In Van Nil experiment equation, what does H2 stands for? Hydrogen donor. In later experiment confirmed that Van Nil data assumption comes from water. Hill's experiment. 5 minutes break. Site of photosynthesis. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. Site for photosynthesis. Photosynthesis occurs in chloroplast. Photosynthesis occurs in chloroplast, which are mainly found in the mesophyll cells of leaves. Structure of chloroplast: outer membrane, inner membrane, stromal lamella. Structure of chloroplast with marking. Outer membrane, inner membrane, granum, thylakoid, stroma lamella. outer membrane, inner membrane, granum, thylakoid, stroma, stroma lamella, granum, thylakoid, stroma, breathe is the called stroma lamella. So next is outer membrane, smooth, permeable to small molecules. Smooth, permeable to small molecules. Inner membrane, selectively permeable, regulates transport. Stroma, the fluid filled space inside site of Calvin cycle. Stroma is the site of Calvin cycle and contains enzymes, DNA and ribosome. Contains enzyme, DNA, thylakoid, disc-like structure stacked to form grana. Disc-like structure stacked to form grana. Site for light-dependent reactions. Contains chlorophyll pigments, electron carriers. Grana stack of thylakoids where light is absorbed. Integral lamella connects between the grana. Summary: thylakoid, light dependent reaction; stroma, light independent reaction. Calvin cycle. So outer membrane is smooth, permeable to small molecules. Inner membrane selectively permeable, regulates transport. Stroma, the fluid filled cavity space inside site for Calvin cycle, light independent or dark reaction. Contains enzyme or DNA and ribosome. Thylakoids, disc-like structure stacked to form grana. Site for light dependent reaction. Contains chlorine. So stroma is site for independent light reaction, that is site of Calvin cycle. Contains enzyme, DNA and ribosome. And thylakoids, disc-like structure stacked to form grana. Site of the light dependent reaction. Contains chlorophyll, pigments and electron carrier. Contains chlorophyll, pigments and electron carrier. Grana stacked of thylakoids where light is absorbed. Intergranal lamella connects between the grana. Thylakoid, light reaction of photosynthesis and water, mainly pitch formation. Stroma, light independent reaction. That is the difference. What is the main site of photosynthesis in plant cell? Chloroplast in the mesophyll cells of the leaves. Which part of the chloroplast contains chlorophyll pigment? Thylakoid membranes in the grana. Where do light reaction depend occur in the chloroplast? Only on the thylakoid membranes. Where do light independent reaction? That is stroma of the chloroplast. What are grana? Stacks of thylakoids inside the chloroplast that contain chlorophyll. What connects different grana in the stromal lamella, also called integral lamella? What is the function of stroma? It contains enzyme for the Calvin cycle as well as DNA, ribosome and other materials. What is the main site of photosynthesis in plant cell? Chloroplast in the mesophyll cells of the leaf. Chloroplast in the mesophyll cell of the leaf. Which part of the chloroplast contains chlorophyll pigments? Thylakoid membranes in the grana. Thylakoid membranes in the grana. Where do light-dependent reactions occur? On the thylakoid membranes. Where do light-independent dark or Calvin cycle takes place? Stroma. What are the grana? The strands of thylakoids inside the chloroplast that contain are called grana. Contain chlorophyll. What connects different grana? Stromal lamella, also called intergranal lamella. What is the function of the stroma? It contains enzyme for the Calvin cycle as well as DNA, ribosome and other materials. Done. Next topic is pigments involved in photosynthesis. Pigments involved in photosynthesis. Chlorophyll A, C55H72, oxygen 5, nitrogen 4, magnesium. Bright or blue green in chromatogreen. Absorbs blue and red light. Role directly involved in light reactions of photosynthesis. Chlorophyll B, C55H70O6, N4, magnesium. Yellow green, accessory pigment. Absorbs blue and orange. Transfers energy to chlorophyll A. Absorbs blue and orange. Xanthophyll. Major difference between chlorophyll A and B is that methyl group of CHL A is replaced by the aldehyde group in CHL. Chlorophyll B is chloro. A is replaced by the aldehyde group chloro B. Xanthophyll yellow accessory pigment absorbs blue light. Carotenoid yellow to yellow orange. Carotenoid another carotenoid color orange and absorbs blue and green light. Anthocyanins water soluble pigment ratio of xanthophyll and carotene in plant is 2:1. Quanta sum occurs in the surface of plasma lemma. Pigments are responsible for light absorption from sunlight at specific wavelength. Most of the processing takes place in the blue and red region of the spectrum. Green light has no role in photosynthesis process. Another carotenoid color orange. Functional SI pigment broaden and absorption spectrum. No sunlight used. Protect chlorophyll from photo damage by dissipating excess energy. Absorption of spectrum versus action spectrum shows which wavelengths each pigment absorbs. Action shows which regions are most effective for photosynthesis. Peaks in blue and red region. Okay. Hello bro, CR7. Pigments involve chlorophyll A bright or blue-green in chromatogram, chlorophyll B yellow-green accessory pigment. Major difference between chlorophyll A and chlorophyll B is the methyl group of chloro A and chloro B. Xanthophyll yellow accessory pigment. Xanthophyll a type, okay. Color is yellow, absorbs blue light. Okay, chlorophyll B absorbs blue and orange. Chlorophyll A is blue-green, absorbs blue and red light, role directly involved in light reactions of photosynthesis. Chlorophyll B accessory pigment. Primary pigment is chlorophyll A. Accessory pigment is chlorophyll B. Color yellow-green, absorbs blue and orange light and transfers energy to chlorophyll A. Xanthophyll a type of carotenoid. Color yellow, absorbs blue light. Carotenes another carotenoid. Color orange, absorbs blue and green light. Okay. Green light has no role in photosynthesis. Most of the photosynthesis takes place in blue and red regions. So absorbs blue and red light. तो कुछ पड़ जाओ flashcard photosynthesis. What is the primary pigment involved in photosynthesis? The primary pigment involved in photosynthesis is chlorophyll A.
में जाना है और इसी साल जाना है। नीट 2025 की बेटा तैयारी कर रहे हैं, शानदार तरीके से बेटा आगाज हो चुका है, बहुत धुआंधार तरीके से बेटा आपकी क्लासेस चल रही हैं। बेटा आप लोग रेगुलरली सारे के सारे सेशंस दे रहे हैं। मैंने आप लोगों को बस एक चीज बोली है कि आप 3 घंटे दिन के मुझे दे दीजिए, है ना, सिर्फ 3 घंटे आप लोगों को मुझे देने हैं बेटा। और केमिस्ट्री के लेक्चर भूल जाइए, फिजिकल केमिस्ट्री हो, इनऑर्गेनिक केमिस्ट्री हो, ऑर्गेनिक केमिस्ट्री हो, सारी चीजें बेटा आप लोगों को मैं करवाने वाला हूँ अलॉन्ग विद द क्वेश्चन प्रैक्टिस। बेटा ना क्वेश्चन प्रैक्टिस में बेटा ढेर सारी करने वाले हैं और प्रहार सीरीज में आप सभी लोगों का स्वागत है। डे 5 पर देखिएगा आप ना कितने दिन की सीरीज है ये प्रहार सीरीज। अगर मैं आप लोगों को बताऊं तो सिर्फ और सिर्फ बेटा 5 दिन की है। इसमें सिर्फ और सिर्फ हम लोग 5 लेक्चर लेने वाले हैं। लेकिन जिसने बेटा ये 5 लेक्चर देख लिए है ना और जो मैं बोल रहा हूँ उसको फॉलो कर लिया, तो आज क्या है बेटा मंडे, 5 दिन कब होंगे हमारे, फ्राइडे को होंगे। आप लोग खुद आके मेरे को बोलोगे कि सर कॉन्फिडेंस सुपर डुपर हाई है। आपने जो बोला बस अपने को शिद्दत के साथ फॉलो करा। ये वीडियो डाली और उसके कुछ ही देर बाद कुछ ऐसा
Which photosystem function first in light reaction? Photosystem 2. What is the reaction center of PS1 called? P700 absorbs light at 700 nm. Which photosystem is responsible for the photolysis of water? PS2. What is the main function of PS1? To produce NADPH. From where does PS2 get its electron? From water. Is oxygen released by PS1 or PS2? PS2 release oxygen. Which photosystem has a reaction center of P680? In which photosystem does photolysis of water occur? Photolysis of water occur in PS2. What is the primary role of photosystem? Primary role of photosystem 1 is formation of NADPH. From where does PS1 receives its electrons? From PS2. Which photosystem works first in the light reaction pathway? Which photosystem work first in the light reaction pathway? 1, 2, 4, 5. 3C. ओके सो आज जो हमने पढ़ा अप टू लाइट रिएक्शन पढ़ा। इससे पहले मैं एक बार आपको सारे जो फ्लैश कार्ड्स है एक बार रिवाइज करके हम लोग आज छुट्टी इस फोटोसिंथेसिस चैप्टर से लेने वाले हैं। ठीक है, चलिए। What is the aim of the half-leaf experiment? To prove that carbon dioxide is essential for photosynthesis. What is the plant kept in darkness before the experiment? To discharge the plant, remove any pre-existing starch. Why is the potassium hydroxide used for this experiment? KOH absorbs carbon dioxide from the air inside the flask. What happens when the half of the leaf inside the flask? It does not turn blue-black with iodine because it did not receive carbon dioxide, so no starch was formed. What happens to the half leaf of the inside the flower? What happens to the half of the leaf outside the flask? It turns blue black with iodine as it got CO2. So called. What is the conclusion of the half leaf experiment? Half leaf experiment conclusion is that CO2 is released from for photosynthesis, essential for photosynthesis. Fasclar Joseph Priestley. Priestley observed that to find out the plants how affect the air around them. What happened when Priestley placed a candle? The candle flame out after some time due to lack of oxygen. What happens when a mouse was placed in a closed bell jar? The mouse died due to suffocation. What change did Priestley observe when a mint ordered the candle burned again and the mouse survived longer? Ha! Key! What was Priestley's conclusion from his experiment? Green plants restored the air. They release something that necessary for breathing and burning. He showed that plants purify air by releasing oxygen as a key step forward. Flashcard site for photosynthesis. What is the main site of photosynthesis in plant cell? Chloroplast in mesophyll cells of the leaves. Which part of the chloroplast contain chlorophyll pigments? Thylakoid membrane in the grana. Thylakoid membranes. Where do light reactions occur in the chloroplast? On the thylakoid membrane. Where do light independent reaction occur? In the stroma. What are grana? Stacks of thylakoid inside the chloroplast that contain chlorophyll. What connects different grana in a chloroplast? Stromal lamella. What is the function of stroma? It contains enzyme for the Calvin cycle as well as DNA, ribosome and other materials. For flashcard for photosynthetic pigment. What is the primary pigment involved in photosynthesis? Chlorophyll A. What is the color? What color is chlorophyll A and which light it absorbs? Blue green is the color and it absorbs blue and red. What is the role of chlorophyll B in photosynthesis? Acts as a necessary pigment transferring energy to chlorophyll A. Name two types of carotenoids in plants. Xanthophyll is yellow and carotenoids is orange. Orange and carotenoids are absorbs blue and green light, and the absorbs what type of light do carotenoid absorb? Blue and green light. What is the function of accessory pigment in photosynthesis? They broaden the absorption spectrum and protect chlorophyll from photo damage. What is the function of accessory pigment? They broaden the absorption spectrum. And okay, what is the difference between absorption spectrum? Shows light absorbed by pigment. Action spectrum shows light most effective in the driving photosynthesis. Light reaction. Where does the light reaction of photosynthesis takes place? Light reaction of photosynthesis takes place in the thylakoid membranes of the chloroplast. What are the main products of the light reaction? ATP, NADPH and O2. What is the process of breaking water using light called photolysis? Breaking of water using light. Which two protein complexes are involved in light reaction? PS1 and PS2. What is the function of light harvesting complex? They absorb sunlight and transfer energy to chlorophyll A. Difference between PS1. Which photosystem function first in light reaction? PS2. What is the reaction of PS1 called? P700. Absorbs 700 nanometer of light. Which photosystem is responsible for photolysis of water? Photosystem 2, PS2. What is the main function of PS1 in the light reaction? To produce NADPH. From where does PS2 get electron? From water. In oxygen, PS1 and PS2. PS2 is oxygen. So done and dusted today. Electron transport chain is there. You have to read tomorrow. Where the it may be used. Calvin cycle, back, photo respiration. So 1, 2, 3, 4, 5, 6. There are 6 topics for tomorrow. Thank you and have a nice day.